Hi, my name is Maria Contreras, and I'm the new Director of Research here at the Masonic Medical Research Institute. Welcome to our laboratory. I'm really excited to give you a tour of our laboratory today, where we've spent the last year doing renovations on the second and third floor, as well as to show you the basement level where we have genetics and additional research that will incorporate many of the efforts that we're making to combat cardiovascular research. Welcome to the genetics wing. This space was renovated about 10 years ago to encompass state-of-the-art sequencing facilities that will allow us to identify new genes that are involved in cardiovascular arrhythmias and sudden cardiac death. We have sequence analyzers, we have data that will allow us to do analysis for all of the DNA from either human patients or animal samples that can incorporate some of the work that we do upstairs on the second floor at the molecular level to understand disease etiology that causes cardiovascular disease and risk. Welcome to the second floor molecular biology suite. Here we have an area that will allow several investigators to focus on their various interests in disease biology of the heart, as well as facilities that can help incorporate many of their research efforts, which include the tissue culture room, the microscopy suite, as well as the chemistry space. Let me show you. Here we are in what will become the future microscopy and imaging suite for the laboratory. Here we will have the capacity to visualize cells within the culture that have movement for cardiomyocytes, as well as be able to visualize fluorophores that can target to specific regions within the cardiomyocyte or the heart cell in order to delineate specific disease etiology. We hope to have a Keyence microscope in this space, as well as a confocal microscope, to be able to do these research efforts. This room was kindly supported by the Royal Arch Masons, who will be dedicating this room to the laboratory at the end of July. Here we are in our brand new tissue culture suite. Here we have the capacity to study primary cells isolated from small animals, mice and rats, as well as to do tissue culture in established cell lines to be able to understand disease biology in a much more informative way. We also do adenoviral, lentiviral delivery of drugs to these cardiomyocytes and other cell types in order to understand the disease in a much better way. And finally, we also do inducible pluripotent stem cell research here, where we can take blood cells from human patients and turn them into cardiomyocytes and culture them here into this room. This is our chemistry suite. Here we store and use our chemicals in order to combine and make molecular biology tools that can help us understand cardiac disease in a more informative way. Jason McCarthy's lab has already gone ahead and made reagents that can help us target disease to combat fibrosis or dying of the specific heart cells, as well as other disease etiologies that control heart failure and disease. We have also created a full walk-in 4-degree refrigerator that will allow us to do research in a cold temperature to be able to understand molecular biology tools and techniques to get to the disease core of cardiovascular heart failure. As you can see, we have ability to store dewars of liquid nitrogen in here, as well as use tools that will allow us to do Western blots and immunoprecipitation experiments to understand protein biology that controls cardiac disease. The design of this space was created to build space for 6 to 7 investigators, along with 4 to 5 postdoctoral fellows, students, and technicians who work within those laboratories to work collaboratively and to network to create an opportunity to come together and build and find cures for cardiac disease. Welcome to our third floor. This is our full barrier animal vivarium, where we keep mice and rats in our facility. I'm happy to show you a little bit about what we do here in this facility and how important this kind of research is for undertaking cardiovascular disease and research efforts. This is an example of one of our housing rooms. This has the capacity to store several cages of mouse genetic models that allow us to do research on the second and genetics floor wings of the building so that we can better combat cardiovascular disease. We have state-of-the-art full barrier space within this room which allows us to feed and water the animals directly from the cage racks, and allow us to be able to better handle and control for temperature, food intake, as well as other parameters that allow us to do research here. Many of these genetic models actually incorporate not just cardiovascular disease, but diseases that include diabetes, obesity, autoimmune disease, and gastrointestinal diseases as well. Welcome to our third floor procedure room on small animal models like mice and rats. Here we have a state-of-the-art echo machine called the Vivo 3200. It is the same equipment that we use in human patients to do echocardiographs on disease model systems to understand the function of the heart. We can apply the same mechanisms to understand the degree of cardiac disease in each particular genetic mouse model that we study. The probes for this are a little bit smaller than what you would normally see in a human patient, but sufficient enough to be able to measure the degree of cardiac disease. Over here, we have a micro-CT scanner. This is the equivalent of a CT scanner that you would use in a human patient. We are allowed to do then live imaging on patients, or in our case, mouse models, that allow us to understand in real time what's happening to the cardiac function in these particular animals. Finally, we have what's called an IVUS spectrum piece of equipment that allows us to do live imaging of fluorophores that travel through the heart in order to be able to determine if therapeutic agents that we're using to target specific disease actually reach their destination and have a therapeutic efficacy. Together, these pieces of equipment are in the order of $1.5 million and constitute one of the biggest contributions of the Masonic Medical Research Laboratory to our research efforts. Here we are in one of the facilities rooms on the third floor vivarium. You can see behind me we have a cage wash that allows us to run the dirty cages from the other side of this room through this cage into what we call a sterile hold, which is where we currently are standing. In addition, we have the opportunity to autoclave all of the reagents and the bedding, as well as the cages that come through from the dirty side to the clean side, so that when we move them through to the other side of the room here, we'll be able to facilitate having clean and sterile cages for all of our animals. We have PHS assurance, and we're currently trying to get our ALAC licensing so that we have the best possible standards for our animal care here at the Masonic Medical Research Institute. Thank you for joining me on our tour today. I hope you've enjoyed looking and learning a little bit about the research that we do here at the Masonic Medical Research Institute. We hope to see you here in person, and I'd be happy and delighted to give you a personalized tour of all of our research here at the lab. Thank you and have a great day.